Today we're talking about using affinity purification to purify antibodies. In order to give you a good overview, I'll talk a little bit about affinity purification in general and then discuss some important structural features of antibodies because that's relevant to how affinity purification works when it comes to purifying antibodies. And finally, we'll learn about the different affinity proteins used and how to decide which one is best for you. So to get started, let's just go over affinity chromatography, otherwise known as affinity purification. Affinity purification is one of the most commonly used strategies in the lab, and often other subsequent purification approaches are used after affinity purification. Let's just imagine you have a mix of proteins and molecules, and you're trying to separate out a specific type of protein from all the other proteins and molecules. With affinity purification, you're separating molecules based on binding qualities. Some examples are antigen and antibodies, enzyme and substrate, and receptor and ligand. When we think about an enzyme and substrate, a substrate will bind very specifically to a certain site on an enzyme. The same goes with antibodies and binding sites or epitopes of an antigen. These relationships are abundant in nature, and the same principles are used by researchers in order to isolate and purify biomolecules. So now we need to talk about antibodies and specifically antibody structure. Antibodies are Y-shaped molecules made of 4 proteins, 2 heavy chains and 2 light chains. These chains are connected by disulfide bonds, giving the antibodies their distinct shape. The top of the Y has variable regions that bind to specific targets or antigens, and at the base of the Y is the constant region, which interacts with other parts of the immune system. There are 5 major types or classes of antibodies: IgA, IgD, IgE, IgG, and IgM. Each class has different roles and is found in distinct locations within our bodies. For example, IgA is common in mucosal areas like saliva and tears, while IgG is the main player in blood and extracellular fluid. Here's a quick rundown of these different classes. As you can see, each one has unique features that make them specialized for different tasks in the body, but this diversity is also part of why antibody purification can be complex, which I'll get into very soon. Okay, so now let's move into the affinity proteins. For antibodies, there are 3 common affinity proteins: protein A, protein G, and protein L. Protein A comes from the bacterium Staphylococcus aureus and binds to the constant region of antibodies. It's particularly useful for purifying IgG antibodies from human, mice, and rabbits. The next protein is protein G, which is a surface protein found on certain types of streptococcal bacteria. It binds to antibodies similarly to protein A, but with a broader range. It becomes a great choice for purifying antibodies from goats, sheep, and cows. Just watch out though, because some forms of protein G can bind to serum albumin, which is a super abundant protein that might interfere with your antibody purification. And then we have protein L. Unlike protein A and G, protein L binds to the light chains of antibodies, not the heavy chains. This makes it ideal for working with IgA, IgD, and IgM classes in human, mouse, or rat samples. But it won't work for every antibody type, so you'll want to look at the compatibility overall before using this one. We just briefly mentioned which proteins are best for each antibody type, but I want to recap this and give you a quicker guide here. Protein A is best for IgG antibodies from humans, rabbits, and certain other mammals. Protein G is good for IgG antibodies from goats, sheep, and cows. Protein L is good for IgA, IgD, and IgM antibodies from a variety of species. I'll also give you plenty of resources in the description to help you out. But again, looking over your lab's protocols and approaches and confirming what is most appropriate is also a really important step in deciding. Affinity purification is a really powerful technique, but selecting the right protein is key to getting the best results. Keep in mind that every protein has its strengths and limitations, so always consider your specific application and antibody type when choosing your purification strategy.
If you're interested in other resources to help you along, you might want to check out GoldBio's handbooks. Just visit goldbio.com/handbooks or check out the link in the description to browse and download.